Well, hello everyone. I'm really glad to be here, to see everyone. I'm a microbiologist, so I study microbes in extreme environments, so deep sea vents, but also deep geological reservoirs. And today I'm going to talk about how we explore the life in extreme environments and focus on the deep sea vents. So I know that not all of you are really familiar with biology, so I will start with some basics in microbiology and then introduce what is an extremophile. Then I will focus on one extreme environment, so the deep sea vents. And then I will present to you how we explore deep sea vents. So I was part in a oceanographic cruise in the mid-Cameron Rise the beginning of 2020. And then I will eventually conclude with the perspectives in the field. So let's start with the basics. So let's start really simple. So what are microbes? So microbes are really tiny cells. We are talking about micrometer scale. So in the microbes, we can talk about bacteria, archaea, viruses, algae, yeast, and small animals. So they are really tiny and you cannot see them with your eyes. You need a microscope. We have different types of cells in the microbes. We have the prokaryotes and the eukaryotes. So prokaryotes are really simple cells. They don't have a nucleus. And they are usually smaller than eukaryotes. So eukaryotes and prokaryotes share similar characteristics. So they have flagella to move in the media. They have a membrane, so to exchange with the surrounding environments. They can divide to grow and multiply the number of cells. They have a cytoplasm, so where you find all the genetic materials and so on. They have ribosomes for protein synthesis and chromosomes, so like the DNA. So each type of cell has a unique set of distinct properties. And in the prokaryotes, we talk about bacteria and archaea. In the eukaryotes, we have different organisms, protista, plants and animals and fungi. And we know that even if archaea and bacteria look the same, they are not closed genealogy speaking. So archaea are more closely related to eukaryotes. And that's what we see on a phylogenetic tree. Like here, so that's the tree of life. So here you see all the diversity of bacteria. And down there, eukaryotes and archaea, which are closely related compared to bacteria. So here, which is interesting, is that the bacteria, it's a large diversity, and it's the 2/3 of Earth's life on Earth. So if you wonder how a microbe look like, under the microscope you can find different shapes. So you have basically some coccus, bacillus, vibrio, coccobacillus, spirillum, spirochete. And sometimes you can find unusual shapes like star shape and also triangular or square shaped cells. And they have different morphology, and they can aggregate together or form some long chains or some grafts. So here is again the size of bacterium. So you see that they're really tiny, and so you need a microscope to see them. And how does a microbe work? So the microbial metabolism relies on carbon sources, so all the nutrients, and energy sources. So the cell needs to find carbon sources and energy from the environment. So the carbon sources, we call this metabolism anabolism. It's the biosynthesis of all the macromolecules in the cells, so all the building blocks and the cellular components. And the energy is the catabolisms, so it's gonna be used for the biosynthesis, but also for other cell functions such as mobility, growth, transport. And as you know, so they produce energy, they produce some components, and also waste products. And we are using this waste, notably with wine or fermentation with beer. So microbes are really useful for humans and not only pathogens. So when you think about microbes, don't think about pathogens at all. They can serve for us, for example. So the microbes really interact with the surrounding environments. So when you think about a microbe, where it lives, you have different carbon sources. So in microbiology, we call them heterotrophic when they need really organic compounds, like glucose or bigger molecules. And we call them autotrophs when they need inorganic compounds. For the energy source, you have the photosynthesis, so the energy from the light, and also energy from all the chemicals, and we call them chemotrophs. So all of this, the microbial metabolism relies on redox reactions with acceptors and donors of electrons. But microbes do not need only carbon sources and energy to grow. There are other growing considerations to take into account, such as the temperature, the salinity, the pH, the oxygen concentration, and the pressure. And if you think as what we usually see around us, we can think that microbes only grow between medium temperature, low salinity, neutral pH, oxic conditions, and at a low pressure, atmospheric pressure. But that's a human point of view, because we know that microbes can grow at a really big range of temperature, salinity, pH, oxygen concentration, and pressure. So here is an example of the temperature. So mesophiles are all the microbes and other animals living in medium temperature conditions. But you also have psychrophiles, which love cold temperature, thermophiles, and hyperthermophiles for heat-loving microbes. So we call those condition as anthropocentric because there's a human point of view, but it's really different from what microorganisms can really love. So we know that microbes can colonize different environments, even if you think that it's unable to find any life there. But if you look closely, you can find tiny, tiny cells, and they can live and thrive in really extreme environments. So here is a picture of different extreme environments on Earth. And all those environments are hostile for life as we know it, but extreme organisms, extremophiles, live in there. So who are those extremophiles? So considering the temperature, we can find psychrophiles, so the cold-loving microorganisms. So they usually grow better at temperature below 15 degrees C. And here is an example of Poloromenas vacuolata. It was isolated from the Antarctica. At the other extreme, you have thermophiles and hyperthermophiles. So they grow well at between 60-80 degrees, and hyperthermophiles below 80 degrees C. So here's an example. So Pyrolobus frommari was isolated from a deep sea vents. Concerning the pH, we have the alkalophiles for very low pH, so a very high pH, sorry. So they grow at pH over 9. And here is an example with Natronobacterium gregoria, which was isolated from a soda lake. And at the other extreme, we have acidophiles with really low pH, so below 3. And Picrophilus oshimae was isolated from geothermal vent. Considering the water activity, we have all the xerophiles. So those microorganisms can grow in really, really dry conditions without any water. So you have Streptomyces here. And you can find also those organisms in like really high sugar concentration. And you have also halophiles and hyperhalophiles, which grow at really high salinity. And Halobacterium salinarum was isolated from really salty lake, can grow up to almost the saturation of sodium chloride. We can also find some radio-resistant microorganisms. So they can cope with really UV and gamma radiation. So here is an example with Deinococcus radiodurans. So it's a well-known bacteria which can cope with really high radiation. So 5,000 gray, which is 500 times the dose of radiation that a human can live with. And you also have piezophiles, so the one that live under high pressure conditions, so above 20 MPa, which is 200 times the atmospheric pressure. And here is an example with Colwellia. So this bacteria was isolated from the deeper point on Earth in the Mariana Trench, so the Challenger Deep, at 11,000 meter depths. And you can see that it can grow more than 1,000 times the atmospheric pressure. So how do those microbes do for living in these extreme environments? So they develop different strategies to cope with those extreme conditions. So here you have a cell with different extremophiles. And all the strategies we can find, for example, chaperones for thermophiles, so to cope with really high temperature or also cold temperature, and the chaperones are going to help the proteins to stay in the good native state and to be stable and not to be unfolding, because when they are unfolded, the bacteria cannot synthesize other proteins, for example, or other cannot metabolize. So you have also some DNA repair strategies. So you can find this in UV resistant bacteria or also thermophiles. So it's a mechanism where the DNA, when it's degraded by all the extreme conditions, the cells have some DNA repairing for really fast to avoid the DNA degradation. You can find also antioxidants. In thermophiles and also psychrophiles, you can find some cells, so they're going to adapt the membrane compositions, so to keep a fluid membrane. And it's really important because all the process with the transport, with the nutrients when they look for food is about works with the membrane. So it's really crucial for the cells to maintain a fluid membrane. So they adapt the lipid composition of the membrane to keep the membrane fluid. You can find also microorganisms which are accumulate organic solutes. So those molecules act as osmoprotectants. So you can find that in the halophiles, for example, and also some psychrophiles, and also acidophiles. So, which is really important here is to think that acidophile, for example, and alkalophiles keep the pH in the cells neutral. So they have developed mechanisms to keep the pH neutral. So for example, alkalophiles have a specific transporter on the membrane to avoid the entrance of hydroxide sodium ions. And they're gonna favorize the entrance of protons to acidify the cytoplasm. On the contrary, acidophiles will maintain a neutral pH in the cell, in the cytoplasm, with potassium, with other ions, potassium accumulation. In the UV resistant microorganisms, you also have DNA repair and antioxidant, and the cells that live under really dry conditions, so the xerophiles, can also develop sporulation mechanism. So it's a really well-known mechanism for bacteria. When they are under stress conditions, they're gonna sporulate. So it's a kind of a dormancy state where all the metabolism is stopped, and this condition allows the cells to be really resistant to extreme conditions. And when the conditions are more favorable, they start forming cells that can grow again and can form other spores when it's going bad. And also UPS production in the xeric xerophiles. And so here those extra polysaccharides are gonna keep the water for dry conditions. So here we don't have the piezophils, but I'm going to talk about it. So what are the specific mechanisms developed by piezophils to grow under high pressure conditions? So they also develop some adaptation with the membrane. So they're going to play with the arrangement of lipid composition to grow as and have a fluid membrane under high pressure conditions, and also accumulate organic solutes to maintain a good protein folding. But what we know about those extremophiles, they are also polyextremophiles. So some of them combine different strategies to cope with multiple stress conditions. So for examples, Natronobacterium is alkaliphiles, but also allophiles. The Eunococcus radiodurans, we call it the Conan bacteria because it's the most resistant bacteria we know, and it can resist also really dry conditions and also vacuum. So some hyperthermophiles and piezophiles, some hyperthermophiles can be piezophiles when they are isolated from deep sea vents, and some piezophiles are psychrophiles when they are isolated from really deep sea and cold deep sea. Some acidophiles are thermophiles, and the hyperhydrophiles are also xerophile, can resist vacuum and also high radiation. So you see that those microbes developed a lot of strategies to cope with different stress conditions, and they can grow in various extreme conditions. So why are we studying extremophiles? So as microbiologists really want to explore the diversity of life, but also push the boundaries of limits of life, and extremophiles are really good for this kind of study. But also we can study extremophiles to know more about the specific molecular adaptations, notably the stability of biological molecules under extreme conditions. And this leads to several applications in biotechnology. So we can find some extremo enzymes. For example, the really well-known Taq polymerase, which is used in PCR. And it's a unique enzyme to do PCR, because during this process to amplify the DNA, you need a special step at really high temperature. And high temperature usually denaturates the most common proteins. So you really need a thermostable enzyme. And the Taq polymerase was isolated from deep sea vent microorganisms. You can also think about extremophiles for sustainable energy, because they can produce green hydrogen, or some Streptomyces species can also produce some antibiotics that are really useful for all the pharmaceutical industry, or because they can cope with heavy metals and high radiation, we can use extremophile for bioremediation of polluted water, for example. And because those extremophiles can grow in really harsh conditions, like it was, for example, on early Earth, we can think about extremophile with the origins of life, and maybe on other planetary bodies. So now you know more about extremophiles. I'd like to focus on one extreme environment, which are the deep sea vents. So first of all, where are they? So you can find deep sea hydrothermal vents all over the globe. So they are mostly under water volcanoes, under spreading ridges and convergent plate boundaries. So here, the well-known Galapagos site was the first discovery of hydrothermal vents in the late 70s. So it's pretty recent. US scientists went to this site to study volcanisms, and they discovered a huge diversity of life, and they were really surprised because they thought that under deep sea conditions there was no life because it was cold, dark, without food. But they were really surprised to discover life in these deep sea vents. And I'd like to talk also about the mid-Caymanian rise, which is the deepest hydrothermal vent known so far, which is at 5,000 meter depth. So you see it at, we have, so all the circles are older discoveries of deep sea vents, and the stars are really recent discoveries of deep sea vents, and we are still looking for deep sea vents because we don't know very well the ocean floor. As an example, we know only 5% of the ocean floor on Earth, and we know better, for example, Mars or the moon surface. So we still have some work to do to better know the ocean floor. So how does the deep sea vent form? So the cold seawater percolates through the ocean crust, and in contact with magma is going to be really hot, and it's going to go through rocks, different rocks, and it's going to be charged with reduced minerals. And in contact with the cold deep sea water, when it's going out, the fluid is super hot and charged with reduced minerals in comparison with the cold deep sea water, which is oxidized water. And these conditions really favor the chemoautotrophy of microorganisms because you can perform some redox reactions. So in the deep sea vents, you have no light at all, so that's why it really relies on the chemosynthesis. So you have thermal gradients, cold and hot, between the fluids and the deep sea water, but also chemical gradients with the oxidized water and the reduced fluids, but also really high pressure conditions. So we can go up to 5,000 times the atmospheric pressure. And these high pressure conditions prevent the water to boil. So those deep sea vents are self-contained ecosystems, and here the chemoautotrophy of microbes sustains all the heterotrophy and all the other life we can find in the deep sea vents, like the macrofauna. So we have two different kinds of deep sea vents. So the hot black smokers. So those are really, really hot, and you can find acidic fluids. So the chemistry here is driven by the magma. There are basalt hosted systems. So in those systems, you have high sulfide concentrations, and so the pH is really low, and reduced, and basically you have precipitation of iron monosil. Comparing to Kohler white smoker, which is very famous in the Lost City site, here the chemistry is quite different because you have the serpentinization process, so at very lower temperature. So the fluid here is only between 50 and 90 degrees C. So it's an ultramafic hosted vent with a high pH, so up to 10, and high hydrogen and methane concentration. So, and the chimneys are formed of carbonates, and they can reach really tall chimneys up to 60-meter depth, meter tall. So you have different reactions occurring in those deep sea vents. So metal oxidation, sulfide oxidation, hydrogen oxidation, but also methanogenesis, anaerobic oxidation of methane. And all these redox reactions are supply the energy, all the metabolic energy for microorganisms. So when you reach a deep sea vent, what you can see first are all the macrofauna. And you can see different mussels, the really well-known tube worms, Riftia fasciptilia, and also some snails, so airy snails and balag snails, but also some crabs here. And we know that we have different fauna between the sites, between the geography. So you can find easily some tube worms and snails in the Pacific. And in the Atlantic, you don't have a lot of tube worms, but you have a lot of shrimps, a lot of them, and also some mussels and some crabs. And this fauna really relies on the chemosynthetic microbes because they cannot find all the energy or food they need, and they are in symbiosis with those deep sea microbes. So here is all the microbial diversity we can find in hydrothermal vents. So it's a huge diversity. So you can find all the thermophiles, so they live close to the deep sea vent fluids. And further, you can find also mesophiles. And you can see that they exchange, because they exchange all the redox reaction to grow in these ecosystems. And you can see that autotrophs here in the green are supplying the food for heterotrophs. So those microorganisms form the base of the food chain in those deep sea vents. So in the deep sea vents, you can find polyextremophiles also. Some examples here are the radio-resistant and hyperthermophiles. And those strains are the most radio-resistant hyperthermophilic archaea we can find on Earth. Besides radio-resistant, you can also find some piezotolerant. So they are hyperthermophiles, but also adapted to high-pressure conditions. So high pressure really affect the cellular components, as we know, for microbial surface microorganisms. And usually high pressure affects the membrane, so it gives a really rigid membrane, it dissociates the ribosomes, unfolds the proteins, inhibit the translation of proteins, and also inhibit the mobility of cells. So we use this high-pressure process to sterilize the food, for example, because it kills surface microorganisms. But we know that some microorganisms from the deep sea can cope with high-pressure conditions, and that's what we see here on this graph. So we classify the microorganisms according to their tolerance to high pressure. So the surface microorganisms, piezosensitive and piezotolerant, cannot grow at high-pressure conditions. But piezophiles and obligate piezophiles really need those high pressure to grow. So what are the mechanisms for them to grow at high pressure conditions? So here is an example of how high pressure is really different between subsurface microorganisms and deep sea microorganisms. So you have the most common strain we use in the microbiology lab, E. coli, which is really sensitive to high pressure. And you see that between 50 MPa and 80 MPa, all the metabolism is inhibited, compared to this strain, which is a strict piezophile, where the optimum growth appears at those little pressure. So we know that we have specific adaptation in those deep sea microorganisms. So here in this table, I show you some hyperthermophiles, really well known. So Methanopyrus candleri has the record of temperature of life. So it can grow up to 122 degrees C. You can still find some bacteria can grow at high pressure conditions. Thermococcus biophilus is a model piezophile for microbiologists interested in deep sea vent microorganisms and high pressure conditions. Pyrococcus cyanosi, so I was talking earlier, is an obligate piezophile, and recently the strain Thermococcus piezophilus has the broadest pressure range, so it can grow from ambient pressure to 130 MPa. And you see that as we go deeper, microbes really need high pressure to grow. So it's really important to know how they do that, and to maintain those high pressure to study them, for example. So here I show you what is the adaptive strategies of deep sea vent microorganisms with Thermococcus biophilus. So it's a model piezophile, and in the microbiology lab, there were a lot of study to try to understand how these microorganisms was doing to survive in those pressure conditions, but also thermal gradients. And with different study, we saw that Biophilus was able to modify the composition of the lipids at high pressure, but also according to different temperature conditions. So it was always a fluid membrane. Concerning the motility, we saw that this strain is only mobile at high pressure conditions, and under pressure, atmospheric pressure condition, the strains cannot swim at all. Concerning the proteome, so all the metabolic activity, we saw that all the proteins in the cells are piezophiles. It means that all the proteins in the cells are adapted to pressure. And we saw that the cell was able to accumulate some solutes, some protection only under stress conditions. And those stress conditions are atmospheric pressure conditions. So it means that all the proteome is adapted to high pressure. And concerning the metabolism, we saw that Biophilus was also able to switch its metabolic activity. So it was able to eat some organic compounds, but also switch to very small compounds. And these strategies really show that this strain is really able and cope under extreme conditions found in deep sea vent. So when we know more about those microorganisms living in extreme environments, we can wonder, is high pressure is a key parameter for microbial life. So we know that high pressure represents the largest habitat on Earth, so it's all the deep biosphere, all the ocean on Earth. And we know that in this deep biosphere, the majority of microorganisms live on Earth, so they all live under high pressure conditions. They are all regulated by high pressure, and this pressure enhances the microbial activity. And we know that we missed a lot of piezophiles so far. We need to do a lot of more studies to discover those piezophiles we don't know. So the pressure influences the microbial activities, and this parameter has to be linked with the deep geochemical cycle to better understand the deep sea vent ecosystems on Earth. But not only on Earth, if you want to understand, for example, the origins of life or possible origin of life in icy ocean walls analog, extremophiles are the main microbes you can be interested in. So NASA was really interested in deep sea vent ecosystem because we know that different ocean walls have liquid water oceans, but also thanks to the rocky the rocks on the underground, they can support chemosynthetic life. So NASA really want to improve and do more and more studies on the deepest vents on Earth in order to explore and probe the limits of life we know on Earth, and also shed light on the biodiversity living in extreme conditions. So one good candidate for that is the Jupiter's moon, Europa, and in several years there will be a mission, so Europa Clipper, and this mission has one objective: to assess Europa's habitability and to see if all the conditions are here to prove that this moon can sustain life. Another candidate is Enceladus, so Saturn's moon Enceladus. And in 2015, for example, Cassini flew through the plumes and found hydrogen, and which is known to be a source of energy for life. So Enceladus is also another candidate for life, possible life in solar systems. So now we talked about deep sea vents. I'm going to present you what I've done during an oceanographic cruise to the Mid-Cameron Rise early 2020. So because we are talking about exploration, how do we explore the biosphere, so all the deep sea environments? We have some boats with research and exploration vessels. And we can also use remoted vehicles, but also gliders or some AUVs. So for deep sea vents, we usually use submersibles vehicles. So those are driven with human. And it's really important to focus on how they are rated. So the Nautil in France is rated, can go deep to 6,000 meters. But Alvin, the US ones, can go only down to 4,000 meters. So if you want to go deeper and study like the deepest vent sites on Earth, you cannot use Alvin. So you have to find something else. So Alvin is going to be upgraded soon to reach 6,000 meter depths. But to go deeper, you can use ROV, so remotely operated vehicles. So they are, you have the boat here, relay with a cable. So this cable is connected to the vehicle, and it gives some energy, but also transfer all the information between the vehicle under the sea and the boat. So in France, we use Victor, which can goes down to 6,000 meters, and in the US is Jason. When you want to sample the deep sea vents, you have different kind of samplers, but when you want to maintain the pressure, you don't, well, we have more samplers that maintain the pressure now. In France, we have the IBIS, developed by Ifremer. And in US, the famous IGT, the isobaric gas tight samplers. And those samplers really can maintain the fluid under in-situ pressure conditions during the ascent. So how does it work when you study a deep sea sample? So first, you sample under high pressure conditions. Then you need to transport this sample still under high pressure conditions. Then transfer to the lab, so we can do equi-pressure transfer. And then we have different growth and cultivation techniques to reproduce the high pressure conditions in the lab. So those are stainless steel vessels, so you can do batch cultivation, but also fluid, continuous fluid cultivations. And then you have analysis of those samples ex situ, so you decompress the samples, or in situ under high pressure conditions, for example, with using the diamond anvil cell. And here is the big challenge for deep sea vent samples, is to maintain the in situ pressure. Because if you don't, bad thing happens. Here you have some example of Methanococcus yanachii strains decompressed really fast after cultivating at 26 MPa, and you see that the cells exploded, which means that when you decompress the samples, some deep sea samples, you kill the cells and you cannot look at them anymore because they are dead, and you have some biases induced with decompression. So there are two rules to follow if you want to work with representative samples of deep sea vents. Rule number 1, no decompression from site to lab. And rule number 2, reproduce in situ pressure conditions. So that's what we tried to do in this cruise, beginning of January. So we went to the Mid-Cameron Rise. So it was an expedition with biologists and also geochemists. And here the goal was to study the chemistry in the deepest vent known so far, the Mid-Cameron Rise. So here it's here in the Caribbean Sea. So we were on the Atlantis, so it's a US boat. So the departure was from Panama, and it took almost 3 to 4 days to get to the site, to the deep sea event site. And we focused on 2 deep sea sites. So one is the Van Damme site at 2,000 meter depths, and the Picard site at 5,000 meter depths. So we combine expertise between different labs, a US lab, but also French lab. And here are all the names of the people involved in this cruise. And I was part of the DEEP project, which is a project granted by PSTAR NASA grant. And here the project named DEEP, meaning Detecting Extraterrestrial Piezophiles in Ocean World Analogues, was to answer this question. When you look for life in deep ocean, does decompression change what you find? So the different scientific goals and challenges were to study the microbiology in those deep sea vents, to assess the effects of decompressions on what you see, so the diversity, but also all the metabolic strategies, but also improve our knowledge on the microbial diversity by isolating new piezophiles, by maintaining really in situ pressure conditions. So better understand those high pressure microbes, their coping strategies, and how they were implied in the deep biogeochemical cycles. And to do that, we developed new experimental tools to address these limitation studies relying to decompression biases. So the first thing to do when you are on a cruise is to build a high pressure lab, because when you want to study deep sea microbes under high pressure conditions, you need all high pressure setup. So here the idea is to transfer the high pressure lab on the boat. So this was done during the 4 days, 3 to 4 days to the site. So you can see here high pressure incubation vessels. We can also have here small anaerobic chamber to mimic anoxic conditions and to work with the sample without oxygen. And different also high pressure line for sample filtration under high pressure conditions and sample transfer without decompression. And you see they're all tight because during the cruise it's pretty bouncy. So the first step is to launch JSON. So it was the rov we used to sample the deep sea vents. So you can see here the launching, the sequential launching. So it's really delicate stuff, because the sea has to be really calm, or they are not able to launch JSON. And once it's in the water, you can go to the JSON command room. So you have a lot of screens. You have 3 engineers driving the rovs. And you also have on the boat some TV screen and you can watch what's going on, what JSON sees in the deep sea. So here the project relies on 4 steps. So we focused on a sampling step with different samplers. We focused on the recovery using different pressure conditions. So no decompression at all, fast decompression, and also slow decompression. With the processing of samples, we work on different enrichments and different filtration processes to collect the DNA, and then the analysis of the samples. So let's start with the sampling. So we used different pressure keeping samplers. So the IGT I was talking about, but also the high pressure MIO sampler. So we had both US and French equipment. And those variable volume cells, which are able to sample 2, but really tiny volume, we use them as pressure incubation systems. So here are all the equipments we use for sampling. So in green here, you can find the pressure keeping samples. And we also use the Niskin bottle and the large volume sampler. So it's a sampler developed by Susan Lung to collect a really large amount of fluids, but those ones are decompressed. So we could compare non-decompressed samples versus decompressed samples. So here on the rov, on JSON, you can see all the IGTs, so on the front desk of JSON. And on the bottom, we had the large sampler, and here is Suzanne just checking that all the valves were ready to go before the launching. So when you, so here is an example of the sampling with the IGT. So you see that here is the IGT in the arm of JSON, and JSON is approaching the deep sea vent fluid, so it's here. So you can see a lot of trims everywhere. And on the cannula, on the sampling side, on the sampling hose, you have a thermocouple just to know and check the temperature of the fluids, and to make sure that you sampled the good fluid you want, depending on what you're looking for, thermophiles or hyperthermophiles, for example. And so it takes approximately 15 minutes to sample the whole IGT. And you have several IGTs on the rov, and they can serve for different sites. And we also used, as I said, the MIO sampler. So this sampler is usually used for sampling the deep sea water column. So it's mounted on a CTD, and it goes down on the water column. But here the challenge was to make these available for deep sea sampling with the rovs. So we adapted this sampler, so it was mounted on one arm of JSON, the swing arm, left one. And we put an hydraulic ram to activate the trigger when we were ready to sample. And we also adapted a nozzle, a titanium nozzle with a sampling hose and a T-handle for JSON to be able to go to the fluids. So it was a really technical and challenging process, but we were happy that it worked pretty well. So once we sampled, we had the recovery process. So as I said, we had 3 steps. So no decompression at all, fast decompression and slow decompression. And for the no decompression part, we used a Necri pressure transfer line. So how does it work? So here you have the IGT as, and it's under in situ pressure conditions. You have here anoxic line, just to make sure the link, the transfer line between the IGT and the recipient vessel are without oxygen. And with the pump, we measure the pressure and the volume transferring in the recipient vessels. And on the back here of the vessels, you have back pressure just to maintain all the pressure in all the line. So we can sample the deep sea samples without any decompression. For processing, we processed non-decompressed samples and decompressed samples, as you can see here. And non-decompressed samples were enriched under in situ pressure conditions without any decompression. And for the one that were decompressed, we recompress them and keep them also at ambient pressure conditions. So here we are able to compare different decompression, so no decompression at all, decompression and recompression, that it's almost happen each time when you sample some deep sea vent organisms. And also some non-recompressed samples. So concerning the enrichment, we used the same technique to sample and transfer the sample between the deep sea sample and the recipient for enrichment. So we selected different media for growth. We selected some methanogens, sulfator reducers. And here it's how it looks like. So you have all the incubation vessels here under pressure. So it's at 500 bars, so the deep sea pressure, maintained at the fluid temperature we recorded during the sampling. So here you have the sampler that was inoculate with the sample and enriched with a specific media. For the process when the samples were decompressed, so we recompressed some of them. So we were using batch reactor with syringes. So here it's the most common way to perform microbiology in high pressure lab. Where you use syringes to grow the microbes under high pressure conditions. And so here the idea was to compare the non-decompressed samples, cultures, with the decompressed and recompressed one. And the second step was to process the samples to know more about the diversity. So to do that, we filter the samples to know to study the DNA, all the genomics. And we perform some filtration under high pressure conditions and after decompression. So as I showed earlier, when you decompress faster, you have explosion of cells. So here the question here is if you rupture the cell rupture during decompression, are we missing the nucleic acids and the proteins for omics studies? So to avoid that, we have some high pressure filters to maintain the filtration under high pressure conditions. And to compare decompression, filtration under decompression, and without decompression, we used samples after decompression and sample never decompressed. So here is the setup. So you have the sample under in situ pressure conditions, and the back pressure valve on the back maintain the pressure all along the line. So you push the samples and it filters under high pressure conditions. So you have the sample here and the filter. So it's so you put the filter in the filtration system, you close, and you can maintain the pressure. For the analysis part, so we wanted to look at the different cell compositions, all the biodiversity we obtained after decompression or no decompression at all. Also look at the metabolic strategies and all the biodiversity we have in deep sea vents and how they were coping with decompression or not, and what we were missing when you decompress samples and when you're not. But COVID hit. So we were unable to process the samples right when we come back from the cruise. So the samples stayed for almost 2 years in the fridge, and we are reprocessing the samples because we don't know what we missed during 2 years in the fridge. We have to compare now what was done during the cruise and what we lost during those 2 years of COVID. So I cannot tell you more about this analysis because it's still in process. So this cruise enabled us to answer some questions here by developing a new technique without decompression to have a deep sea sample under in situ pressure conditions. So we want to improve the biosphere exploration. And what was important in this cruise was to share the expertise between different labs and share all the equipments that you can find in all the labs. And that was important because high pressure is a small niche with specific expertise, but when you combine all the expertise, well, you can do good science. And finally, this study has implication for life, all the biosignatures in the subsurface of other solar system bodies, like ocean worlds, like Europa or Enceladus. So what are the perspectives of deep sea exploration? So talking about perspectives, let's start with the overview in the field. We know that high pressure microbial sampling and high pressure growth are limited by budget. When you do high pressure, it's a really expensive equipment. So not all the labs are equipped with high pressure. So most of the lab, when they look at a deep sea sample, they miss the high pressure part because they don't have the expertise and they don't have the equipment to do so. So here the idea is to make this equipment available to all the community, broader range, and it could allow all the scientists interested in the biosphere to access the majority of the biosphere. So we really need to increase the microbial diversity, but because we don't know a lot about them, and it's also because of the high pressure equipment. And to do so, it will give us a better understanding of those microbes and their adaptation strategies. And as I said, so far we only explored 5% of our oceans, so we really need to improve that to better understand those deep sea ecosystems. So how can we overcome these limitations? So we have different strategies going on. Example of what we can do. So as I mentioned, we are able so far to sample the deep sea samples without any decompression, transfer them in the lab without any decompression. But the challenge here is to be able to grow them and characterize them without any decompression. So in the lab, we developed different tools to transfer the deep sea samples in specific tubing, high pressure tubing, where you can see through them and characterize them with spectroscopic techniques without any decompression. And we also developed in the lab some macrophilic chips. So they are tiny microreactors that can hold the high pressure conditions, and you can use them as microreactors, look at them under the microscope. And an example is to use those tiny chips to mimic deep sea vent. So here the idea in the frame of this French project, Eau de Vie, is to study the prebiotic chemistry at deep sea vent conditions. So this project is performed in the lab in Bordeaux by Thierry Tassin and Samuel Mar. So you have an example here of the microchip where it's gonna mimic the chimney. So you can have inject the hot water to mimic the fluid of the deep sea vents, and also cold water to mimic the oxidized cold water. And in the channel here, you can have some reactive chemicals, some reactive rocks, and just to study the geochemical reactions occurring under deep sea conditions. And so here is a picture of those chips. So you see that they're really tiny and then can go through the microscope. And so those high pressure microphilic setup are new lab scale approach to tackle the limitations in leak with in situ investigation in deep sea vents. So they are used mostly for prebiotic chemistry, but we intend to extend them to deep sea microbes. Another perspective in the field is to perform on-field studies. As you know, the best way to understand those deep sea microbes is to go there, but as human we can't do that. So the idea is to have a lab down there. And I'd like to share with you the Batibot project. So Batibot is a rover. It was launched beginning of February in the Mediterranean Sea. So it's going to be down there for several years. This robot has several objectives. So it's going to study the bioluminescence in this Mediterranean Sea. And this project was developed by Christian Tamburini and Séverine Martini in the MIO. So if you want to know more about Batibot, you have a Twitter account where you can see all the discovery and what it sees all the time. So how can we overcome these limitations? So first, by developing new tools for sampling, but also enrichment of deep microorganisms without any decompressions. We really want to preserve all the in situ pressure conditions. The better way to do that is to combine the expertise within the community. We also need to design new equipment. So microphilic is also part of this development because we can mimic multiple environmental factors and really be close to that extreme conditions and combine in situ analysis without any decompression. And the perspective in the field, as I show you, is to develop subsurface laboratories with on-field studies. So yeah, well, I'm done with this presentation. So thank you for your attention.